工作上。 You cannot start with, okay. Good morning. Can everybody hear me?
Yes, yes.
Hello.
Hi.
Of course, we can hear you.
Okay. Good morning from Istanbul. I am Dr. Ekram Aslan. First of all, I would like to say welcome everybody to this meeting. I want to share my screen. Sorry. The topic of today is non-alcoholic fatty liver disease. we will talk about 1.5 hours about this topic. Prevalence of NAFLD is about 25% if we look at the globally. Its incidence is increased with rising level of obesity, type 2 diabetes, and metabolic syndrome. The natural history of NAFLD can change from steatosis to advanced fibrosis, cirrhosis, and hepatocellular carcinoma. So NAFLD is predicted to become the leading cause of cirrhosis, which is requiring liver transplantation, for example, in the next decade. So we will talk about the NAFLD today, and the plan of the program is firstly we will start with Dr. Igor Knязев with pathophysiology of fatty liver disease. Then Dr. Olga Perekudova will talk about the role of gut microbiota in the pathogenesis of NAFLD. And finally, Dr. Nurgul Toktogulova will talk about the burden of non-alcoholic fatty liver disease in Kyrgyzstan. I would like to introduce Dr. Igor Knязев to talk about pathophysiology of fatty liver disease. And I say finally, welcome everybody in this ICMAS Academy meeting. Thank you very much.
Okay, thank you very much for introduction. So can I ask to give me rights of organizer to demonstrate my screen? Yeah, thank you. So hello again. Thank you for this opportunity to express our thoughts, our knowledge in this training program. So my name is Knязев Igor. I'm teacher of pathology in Салымбеков University. So that's what I want to show you. That's why I want to show you our presentation about pathophysiology of fatty liver disease. Okay. One second. Okay, first of all, I want to start with classification of fatty liver disease. So nowadays, classification is based on this fatty liver disease is divided into two parts: is alcoholic liver disease and non-alcoholic fatty liver disease. Today we concentrate our attention on non-alcoholic fatty liver disease. But as nowadays evidences show that alcoholic liver disease and alcoholic fatty liver disease, they have a lot of in common, especially in a portion of non-alcoholic steatohepatosis. Histological features are almost similar. Okay, and non-alcoholic fatty liver disease is classified to simple fatty liver and non-alcoholic steatohepatosis. So before starting to talk about pathology, I want to introduce you the biochemical features of these disorders. So normal biochemistry of fat metabolism. And there are two main pathways how fat occurred in our body. So when I talk fat, it means triacylglycerides or triglycerides, which are present in our body. So we have two pathways. First pathway is consumption of fat through digestive tract, and second is de novo synthesis of fat. So daily intake of fats, so of lipids, going on approximately in a rate of 80 grams in one day in developed countries, and approximately 90% of these lipids is composed of triacylglycerides. And that's why predominantly we'll discuss it like triacylglycerides digestion right now. Digestion in oral cavity and stomach of fat is insignificant because okay, in the next diagram it will be visible. Predominantly we concentrate our attention on fat metabolism or fat digestion in a small intestine, especially in duodenum. First of all, here we should discuss about the role of bile salt. So bile salts, main function of bile salt, which is released by gallbladder and liver by common bile duct, is to emulsify the lipid droplets. So fat, digested fat should be emulsified split into small droplets, small fat droplets, and make it water soluble, make it possible to form micelles. So then what happens in the lumen of small intestine? Then these droplets, which are formed with the bile juice, they give approach to lipases to digest it. And these triacylglycerides, they can be destroyed, so it can be hydrolyzed to fatty acids and 2 monoacylglycerides. And this and secretion of enzymes, pancreatic enzymes, and bile juice is controlled by secretion of enteroendocrine cells, which produce CCK, cholecystokinin, and also secretin plays a little role in this process. Cholecystokinin, it promotes gallbladder contraction, smooth muscle activity is modulated by CCK, and also they stimulate the secretion of pancreatic enzymes, which one of them is lipase. Okay. So here is a diagram which explains the digestion in the digestion of fat in the digestive tract. So starts in oral cavity, where I said it's not significant for us right now. It's lingual lipases. Their main function is digestion of short and medium chain fatty acids. So which are not in a frame of our topic today. And gastric lipases, so which also serve the same function but in the stomach. And as I said, mainly digestion happens in a duodenum. Finally, when these fatty acids are hydrolyzed by lipases, it forms free fatty acids. And as I said, 2 monoacylglycerol molecules. And together together with cholesterol, which is from there, with the bile salts, they form the mixed micelle. So mixed micelle, it diffuses into the acid microclimate disequilibrium zone, so directly close to the microvilli of absorptive cells of small intestine. So that's how it looks like. Free fatty acid, so chemical structure of free fatty acid, and this MAC, 2 MAC, so 2 monoacylglycerides. So we see glycerol residue, and free fatty fatty acid attached to this by ester bond. Then what happens inside the enterocytes? So how it enters to enterocyte? Free fatty acids, along with 2 monoacylglycerides, they are organized into micelles, and they are absorbed by small intestine. And for this, we use fatty acetyl coenzyme A synthetase, or another name is teokinase. This enzyme attaches this fatty acid, free fatty acid, to the coenzyme A, and this coenzyme A serves to reattachment of this fatty acid, free fatty acid, to monoacylglycerides, 2 monoacylglycerides, and recombining, rejoining of this fat. And you see, in a small intestine, we have a triacylglyceride, then it decomposes in monoacylglyceride, and then it reconstitutes. It forms again the fatty acid, triacylglyceride. Okay, and here I have to know that short chain fatty acids, they do not attach to monoacylglycerides due to its high hydrophility, and as well as hydrophilic, it is directly absorbed by capillaries of the small intestine, which brings this free fatty acid directly to the portal vein. And so in absorption, soluble, they're very large molecules, and that's why they accompany together from a bulge of fat inside the transfer it to the circulation. And for this fate, use the apolipoprotein B48. Apolipoprotein B48 is a product of rough endoplasmic reticulum of enterocytes. It fuses it together and makes it like one bulge structure. The structure name is chylomicron, and here produced nascent chylomicron. This nascent chylomicrons enter into lamina propria of the small intestine, and in lamina propria of small intestine, this chylomicrons, group of chylomicrons after fatty food intake, causes the formation of chyle. There we have lacteals. It's lymphatic. It is a lymphatic capillary which drains the chylomicrons like micelles towards the general lymphatic flow. So, and they're named as lacteals due to its appearance. So it's a milk-like appearance because actually what is milk? Also protein combined with protein which micellizes the fat. Okay, and then this chyle, it's named as chyle. It transports toward the thoracic duct, and from thoracic duct it enters into the left supine vein. Okay, let's repeat it in diagrams. It will be more understandable. So then micelles, they are absorbed by enterocytes, and the monoacylglycerides they go to endoplasmic reticulum directly, and free fatty acids they are with the transporting of fat acid binding protein, so forms a fat acid coenzyme A. This fat acid coenzyme A can be used for beta oxidation, but also today more important for us is the formation of deacylglycerol and triacylglyceride. And these triacylglycerides are collected in lipid droplet, as I said before. Then in rough endoplasmic reticulum produced ApoB48, and this ApoB48 forms a chylomicron. So let's show it. In this diagram explains this when imbalance of glycerol and fatty acid consistence is cleared with the glycerol phosphate pathway. So we will return to this diagram a little later. And here in this diagram, fatty acids are same picture, same idea, but from other angle. So these fatty acids, which are absorbed by enterocytes, they are transported toward the endoplasmic reticulum, and they form triacylglycerides. And in endoplasmic reticulum, it gets attached with ApoB48, which is very important in today's discussion, and ApoA4, apoprotein A4, so which is a little less important in today's discussion. Then this lipid droplet, mainly composed of triacylglycerides, is covered with endoplasmic reticulum membrane, and transporting vesicle facilitates the exocytosis of the cell into the lamina propria below the basal membrane of enterocyte. So where it founds in lacteals, as I said before. So how these chylomicrons transport to liver? So as I said, from the lacteals, it enters to thoracic duct. From thoracic duct, it enters to left subclavian vein, and this nascent chylomicrons transformed with the help of high-density lipoproteins. It gains on the surface two groups of proteins like ApoE, apoprotein E, and apoprotein C. Two types of apoprotein C, C2 and C3. So functions of them, so ApoC2 activates the lipoprotein lipases in muscular capillaries and so skeletal muscles and cardiac muscle capillaries, and also activates lipoprotein lipases of capillaries in adipose tissue. So where this triacylglycerides can be used, where this fat can be used for formation of free fatty acids and using of them in a process of beta oxidation, or in a process of gaining of more fats, a collection of fat in adipose tissue. And apoprotein E, which is responsible for attachment to LPL receptors of hepatocytes, which allows them to be reconstituted, rearranged in hepatocytes. So about this, we'll discuss a little bit. This diagram explains it pretty well. As I said, dietary lipids coming through the small intestines, it enters into the lymphatic system. Then in a bloodstream, it gains, along with the B48, it gains ApoE and ApoCs. Then passing through the capillaries of muscle and adipose tissue, LPL of this lipoprotein lipases of these capillaries degrade these chylomicrons a little bit. And the free fatty acids coming for use to muscle and adipose tissue, and then return the chylomicron remnants. So chylomicron remnants possesses ApoE, apoprotein E, and ApoB48. And because of this ApoE, as I said before, low density lipoprotein receptors on hepatocytes allows it to be absorbed by liver. Production, so we'll discuss a little later. We'll return to this diagram. Fatty acid de novo synthesis. How it goes? So maybe ask me to talk slower. So why we need fatty acid de novo synthesis? First of all, it's due to fat is the most calorie-rich compound, and to store it is the most effective way to store energy in our body. Like carbohydrates and amino acids or proteins, 1 gram of carbohydrates and proteins, it gives only 4 kilocalories. But fat, 1 gram of fat gives 9 kilocalories, and that's why the most effective way to store energy is the fat. Okay, and excessive carbonic consumption, carbohydrates or amino acids, it leads to formation of acetyl coenzyme A. So this process is explained in diagram, next diagram. So this, okay, let's skip. So we have a little time. I will explain in this diagram. So consume glucose will a glucose transporter enter into cytoplasm in the way of phosphorylated glucose 6-phosphate. This glucose 6-phosphate is used for pentose phosphate pathway, as is shown here, or in glycolytic pathway. Glycolytic pathway leads to the extraction of ATP and NADH, reduced nicotinamide diphosphate. It forms pyruvate, and then pyruvate easily enters into the mitochondria matrix. And pyruvate, it has 2 pathways to be digested. It's it's decarboxylation to form acetyl coenzyme A, or carboxylation to get to join with acetyl portion forms a citrate. And actually, I said before, so acetyl enzyme A is responsible for fatty acid secretion. Fatty enzyme A is very large and cannot be penetrated through the inner membrane of mitochondria. So acetyl forms a citrate, water-soluble citrate, and this water-soluble citrate can enter into this, and this citrate in the cytoplasm, so transformed back into acetyl coenzyme A, but cytoplasmic acetyl coenzyme A, and into oxaloacetate. And fate of oxaloacetate is not important today, but fate of acetyl coenzyme A is has very important features now. Acetyl coenzyme A, in this location, it doesn't go directly for fatty acid secretion, fatty acid production, but it uses enzyme, this ACC, acetyl coenzyme A carboxylase, and which transforms acetyl part into malonyl part. And this is a key process for protein synthesis, and it well regulates the fatty acid fatty acid synthesis. Then this malonyl coenzyme A, which is produced by ACC with the help of fatty acid synthase, finally forms a fatty acid. In more details discussed later. So what's the role of ACC in synthesis of fatty acids? Take a look here. So ACC, acetyl coenzyme A carboxylase, as it's shown here on this line. Sorry, my mouse doesn't work properly. Acetyl coenzyme A carboxylase. So it is in active position, but we have a regulator which is named as AMPK. AMPK is a AMP activated protein kinase. This kinase serves to phosphorylation of this acetyl coenzyme A carboxylase, and which converts it in inactive position. And which hormones control it? Of course, it's a glucagon and epinephrine. So, and here we can claim that glucagon secretion and epinephrine secretion causes the interruption of fatty acid synthesis, must be decrease the fatty acid synthesis. And what is on the other side? Of course, it's insulin. So usual antagonist of glucagon and epinephrine. Insulin causes activation of protein phosphatase. Protein phosphatase, which is responsible for dephosphorylation of acetyl coenzyme A carboxylase and converting it into active position. So inorganic phosphate residue left out. Okay. This is a deep biochemical process of fatty acid synthase activity, so which use not only acetyl coenzyme A but also malonyl coenzyme A in synthesis, and gradually, step by step, by the process of reduction of NADPH and dehydration, decarboxylation, and other biochemical changes, leads to elongation of fatty acid chain. And finally, so we here we have butyryl ACP, then here we have hexanyl ACP, and then every step gaining two residues, two carbon residues, leads to formation of palmitate. Palmitate, which is accumulated with ACP, acyl carrier protein. And here, by hydrolysis of this ACP as palmitate, leads to formation of, again, fatty acid synthase and palmitate. So, and this palmitate can be used for it's a free fatty acid, which can be used for triglycerol synthesis. It's a good fatty acid synthase. This we discussed. Okay. A little amount of synthesized fat stores in the liver, but most fat droplets accompanied with other lipids and apoproteins and exocytosed as very low density lipoproteins. Very low density lipoproteins. So let's, okay, here this diagram explains it very well. So liver, in the same manner as in enterocytes. So this fatty acid, fat droplet, a lipid droplet getting attached with ApoB, also ApoB as in small intestine, but ApoB100, and forms a very low density lipoprotein, but it's directed directly into the central vein of the portal of the liver, central vein of the liver, and there it meets same high density lipoprotein, and ApoC and ApoE is attached to the surface of this very low density lipoprotein. So before attachment, it was named as nascent VLDL, but after this, it's a functioning VLDL structure. It's mature VLDL. And usage of this VLDL is similar as in similar as in chylomicrons. Also capillaries, also same apocies. They activate LPL, lipoprotein lipase. And the same free fatty acids are used by muscles and adipose tissue here, and returned the remnant of VLDL or IDL. Then this IDL can be returned directly to liver. Can be used for LDL, low density lipoprotein synthesis. And again, it comes to liver or might be used by peripheral tissues. So you see, cycle is pretty similar. Cycle is pretty similar from small intestine transportation and from liver production of fat. So now let's finally talk about pathology. So we are here for pathophysiology of NAFLD. So causes of NAFLD includes insulin resistance and cellular insults. There are main cellular insults include oxidative stress, lipid oxidation, direct lipid toxicity, mitochondrial dysfunction, and bacterial endotoxins. So about bacterial endotoxins, we have a distinct presentation below. NAFLD, we see NASH. So actually, NAFLD is a group of disorders, which include simple fatty liver and non-alcoholic steatohepatosis. So, and NAFLD, so as was said in the beginning of this presentation by our colleagues, NAFLD so is found in approximately 25% of population. And we term this NAFLD when more than 5% of hepatocytes in a liver are affected with the fatty changes. But NAFLD simple fatty disease, so we have no inflammatory changes. And we talk about NAFLD only in that case when we don't see obvious causes of the other secondary hepatic fat accumulation causes like HCV, hepatitis C virus, Wilson disease, medication, or any other chronic intoxication. In other hand, NASH is a form of non-alcoholic fat liver disease with the histological features of alcoholic steatohepatosis injury. So these pathological features include balloon hepatocytes, inflammation and necrosis, perivenular and pericellular fibrosis. These patients have a greater risk of fibrosis and following cirrhosis. So that will be shown in diagram. So mechanism. So, and here we should say that precise mechanism is unknown. We know that we have strong association with insulin resistance. If patient has insulin resistance, cases of non-alcoholic fat liver disease increase. But severity of insulin resistance is not correlated with the severity of non-alcoholic fat liver disease. So must be we have some other causes, some other predisposition factors for NAFLD progression. So, one of these causes, it is might be related to gut enterotoxin due to composition of gut microbiota, and might be and it is associated with liver inflammation and injury. So more correlated with non-alcoholic steatohepatosis, you see. Then also we see the sleep apnea is associated with NAFLD. So sleep apnea, so we understand it's a chronically intermittent hypoxia state of the patient, and it is related with mitochondrial function injury, mitochondrial function depression, and because of hypoxia, chronic hypoxia. But in other hand, we have to keep in mind that sleep apnea is most commonly present in patients with obesity, and that might be just obesity taken influence to NAFLD, and might be just comorbid state, but not a cause of this NAFLD. So it requires more study. Mechanisms of NAFLD. How insulin resistance is related to this? Insulin resistance is associated with increasing of deliberation of free fatty acids from adipose tissue, and so because of activation of LPL, lipoprotein lipase activity, as we discussed before. So also obesity, which is related with insulin resistance, it decreases the synthesis of adiponectin. It's one of modulator of adiponectin, which is a very good modulator of insulin sensitivity of cells. So it helps to produce insulin receptors. And with obesity, adiponectin level decreases, and then insulin receptors decreases, and that's why it leads to insulin resistance. And this correlation is direct, and severity of adiponectin decreasing is correlated with insulin deficiency, insulin insufficiency, insulin resistance disease, and diabetes type 2, diabetes mellitus type 2. And it is also dysfunction adipose tissue release pro-inflammatory cytokines, and fat-laden cells also, which are related to atherosclerosis. They produce pro-inflammatory cytokines like TNF, like interleukin 6, and it maintains the chronic pro-inflammatory state of the body, which also can injure the hepatocytes in an inflammatory way. Damage of copper cells, it's also by endotoxin, by toll-like receptors, or by major increasing of level of cytokines. It leads to local formation of TNF alpha, which is tumor necrosis factor alpha, and TGF beta, transforming growth factor. Transforming growth factor beta, it causes activation, epigenetic activation of stellate cells, also known as EPA cells of the hepatocyte, and their transformation into fibroblast way, and as fibroblast, they produce collagen in the DC space. And this collagen causes a fibrosis, and it seems like fibrosis there. And later, of course, it can lead to cirrhotic changes too. Then, and of course, if we see that similar insulin resistance we have different clinical features of this disease, must be here we have influence of inherited conditions and epigenetic factors. So from epigenetic factors, so now it's in studying process of many different institutes. It's a role of non-coding short RNA chains in expression of this NAFLD. So one of example of inherited condition that leads to NAFLD, it's a Bassen-Cornswijk syndrome, or another name, abeta lipoproteinemia. It's an autosomal recessive disorder, and which deficiency of microsomal enzymes, which leads to decreased secretion of apoprotein B48 and apoprotein B100 secretion. And as we discussed before, so you remember that this apo B100 helps to transport this lipid droplets from the liver to the bloodstream. And with the deficiency of this ApoB100, of course, this triacylglyceride rise will store in hepatocytes. Okay. Then we have another correlation. It's found that persons with genotype PNPLA3, it's a patatin-like phospholipase domain containing protein. This genotype shows histologic evidences of severity correlations of NAFLD. So severity is correlated with this gene. Exact function of the gene is unclear. Still, we can't determine clearly so what is the role of this gene, but now we know that level of this gene expression is correlated with NAFLD. Other mild epigenetic evidences in laboratory animals show possibility for acquired failure of ApoB secretion by hepatocytes. So in the same way as in beta lipoproteinemia, so it can lead to hepatocyte injury, hepatocyte steatosis. So here, this is diagram. I want to explain. So actually, most of topics, most of points were explained before. So you see, as well as fatty liver disease is related with obesity. So adipose tissue plays one of the key role in involvement. So adipose tissue causes the production of leptin, which inhibits the lipid synthesis. But we have a lot of genetic lesions of leptin synthesis itself or receptors of leptin synthesis that degrades the normal response of the body to this leptin. And that's why it also, this injury of this genetic lesions of these proteins can lead to changes, fatty changes in the liver. Then secretion of interleukin 6 and TNF, like pro-inflammatory responses, as I said before. So it causes activation of TNF receptors, causing the apoptosis and necrosis of liver cells. And of course, it will promote the inflammatory response. And decreasing of adiponectin, as I said before, it's a regulator of insulin receptors, and it promotes insulin resistance. So decreasing of adiponectin causes insulin resistance, which is other cause of fatty liver disease. Okay, this PNPLA3 genotype, we don't exactly understand the mechanism of fatty liver disease, but it is present. Then, from the environment, so which environmental causes we have? It's gut nutrient-derived signals like trans fatty acids. So of course, it's a consumption of food. So with trans fatty acids, so somehow they cause activation of toll-like receptors or other influence. So it's not considered yet, but somehow trans fatty acids causes the liver injury. Fructose intake, increased fructose intake, they can activate TLR, toll-like receptors of enterocytes or Kupffer cells or other cells which maintain the pro-inflammatory state of the liver. And of course, other TLR ligands, which are produced by microbiota of intestine. And these TLR ligands can affect as well as enterocytes, as well as and activation of TLR receptors of enterocytes causes deactivation of PF. PF is a fasting-induced adipocyte factor, fasting-induced adipose factor. So it's which activates the and blockage of PF causing the activation of LPL. Lipoprotein lipases, and which increases the plasma component, plasma level of free fatty acids, which directly cause the oxidative stress in one hand to Kupffer cells and hepatic stellate cells. It's causes the oxidative stress to hepatocytes. And also it's a gain of free fatty acids, which can be used for triacylglyceride secretion inside the hepatocytes. So which together these factors causing the fatty liver disease and non-alcoholic steatohepatosis too. Nowadays, so some studies show that try to differentiate NAFLD and NASH, and they claim that there are two independent disorders, like NAFLD, it's one cause, and NASH, another cause. They don't know where to draw the line. They try to draw this line, and they claim that NAFLD is not a stage of NASH. So simple fatty liver, so it's not it's a predisposition factor for NASH. So that's why, but in this researchers, they claim that they need more investigation, they need more studies, but still this idea is common in scientific society right now. And clinical features. Now, clinical features, so in more details, I think it will be discussed in further reports. What I should say is there are no specific symptoms of NAFLD. So it's primarily by clinicians, it is found usually by ultrasound investigation or blood test results like AST, ALT analysis, where we see elevation. So it's usually used in routine analysis. So symptoms are fit to metabolic syndrome. So death rate is not increased due to NAFLD, but not significantly changed. So in all other patients with metabolic syndrome. And in ASH, you will have to mention that NASH causes a hepatocellular carcinoma and in higher risk range, but it's not so high to change the general mortality. Okay, the goal of treating of patients in this condition is to reverse histological changes in liver and mainly by changing of dietary habits. We have other approaches to treat this disorder, but mainly so it still remains the dietary habit changes. Okay, that's it. My report is over here. Thank you.
Good afternoon, dear colleagues. My greetings to all participants of this meeting. My report is about the role of gut microbiota in the pathogenesis of NAFLD. So, as mentioned in the previous report, NAFLD is the most prevalent liver disorder worldwide. And pathogenesis of NAFLD involves many factors. It involves genetic, environmental, host factors, and factors including those that cause changes in intestinal microbiota. The goal of this report is to show the relationship of microbiota signature with the severity of NAFLD and the role of microbiota in situation with development of NAFLD. At first, I want to speak about such notion, such concept as gut-liver axis. We know that about 70% of vascularization of liver is going from gastrointestinal tract via portal vein. And in this situation, we should speak that our liver is exposed to nutrients, toxins, and antigens from food, to the products of gut microbiota and to microbiota itself. Meanwhile, the gastrointestinal tract receives the product in the form of bile acid, which is channeled through biliary duct. This functional bidirectional relationship between liver and gastrointestinal tract is known as gut-liver axis. It is composed of complex component, and because of close anatomical and functional relationship, changes of one component such as gut barrier and disruption of gut barrier or gut microbiota can affect the positions of change of conditions in liver. And in normal situations, small portion of bacterial cells and gut microbiota and its particles are eliminated by Kupffer cells quite successfully. And in the cases of dysbiosis, the amount of gut microbiota and its product reaching the liver will be greater, and potentially it may provoke tissue damage. So this situation with bidirectional relationship, which is known as gut-liver axis, quite important in our report to explain importance of gut microbiota in progression of NAFLD. So at first, what is it normal microbiota? I like this slide because it shows quantity and importance of our microbiota in our body. We may speak that our body consists of 100 of trillions of human cells and only 100 trillions. But bacterial cells, which we may find in our body, are of 1,500 trillions of microbes. They have its own genome, they have its own function, they have its own metabolic reactions, and its role is quite high. That's why we may speak that microbiome of the host of body, of our body, is really as virtual organ. And to smile a little bit, I see that we may speak that we are really as Petri dish with shoes. So what is normal flora in intestinal tract? In the intestinal, we know that the intestinal tract, microbiota of intestinal tract is beginning to form from birth. And for many years, we were sure that gastrointestinal tract of our newborns is sterile. But now this dogma is also changed. And many researchers showed presence of bacterial cells in tissues of placenta. And I see that in future we may discuss this question and it will be a new way for new investigation. Development of microbiota is finished when we are 3 or 4 years of age. And in general, after 3 or 4 years of age, we may see 3 domains of life in our body: bacteria, archaea, and eukarya. And in this situation, about 200 of dominant species we may find and 1,000 non-dominant species. And of course, these species may be different in different individuals. As a rule, we speak about prevalent situation with anaerobical bacterial cells in our microbiota population. What bacterial cells we may find in normal microbiota? Gram-positive firmicutes, it is the most abundant quantity. Bacteroidetes, which are gram-negative, gram-positive actinobacteria, and gram-negative proteobacteria. Of course, we speak about a situation when our microbiota depends on the site. And in this slide, you see illustration of this dogma. Of course, in the stomach, we may see not so high diversity of bacterial cells, which may be in the last part of intestine. And it is due to presence of strictly acidic conditions of pH, which is about from 5 till 7. And only some types of bacteria may grow, and density of these bacterial cells is not so high. And we see that increasing of number and diversity of bacterial cells is increasing from upper part till lower part of gastrointestinal tract. And it depends on functions also, because if we have pH like 7 from 9 in jejunum, and it is due to presence of fermentation of our food in this and absorption in distal part of intestine. And these positions will explain us presence of different types of bacterial cells. I also wanted to sign that the most part of bacterial cells may be as facultative anaerobes or may be as really obligate anaerobical bacteria. So in the duodenum, we see the most part of Lactobacillus, Enterococcus D, Streptococcus D. In jejunum, we may speak about presence of Enterococcus, Lactobacillus, diphtheroid, and sometimes we may speak about presence of small amount of Candida albicans. In ileum, microorganism begins to resemble to that in large intestine, and we may speak about presence of some species of Clostridium, bacteroids, and Escherichia coli. So this is the normal situation with bacterial cells. And also we should keep in mind that some types of bacterial cells may play a role of anti-inflammatory bacteria. So we may name them as bacterial cells which are healthy promoting bacteria, such as Bacillus fragilis, Eubacterium rectale, Eubacterium hallei, quite important for digestion of carbohydrates, Faecalibacterium proznichi, and bifidobacteria. Pro-inflammatory bacteria, as a rule, these are gram-negative bacteria from species of Escherichia, from Pseudomonas aeruginosa, and from Proteobacterium. What is the role of normal flora in our body? At first, of course, we should speak about maintenance of mucosal barrier integrity. Synthesis of vitamin K and B complex, it is quite famous role about which we know from our student ages. And it is quite important for bacterial cells which are responsible for this synthesis, such as Escherichia coli. Conversion of bile pigments and bile acids, it is also the role of gut microbiota. Of course, these bacterial cells participate in absorption of nutrients and breakdown of different products. Due to presence of potentially healthy potential bacterial cells, we may speak about its antagonism with pathogenic bacteria and colonization resistance to pathogen, due to production of bacteriocins, due to production of lactic acid and change of pH in which gram-negative and pathogenic bacteria may not survive. One of the roles which is quite important, it is constant education of gastrointestinal immune system. It's really a fact which was showed by many scientists, and it is one of the questions of immunological tolerance according to normal flora which may present in our gastrointestinal tract. Regulation of intestinal hormone production such as glucagon like peptide 1 and serotonin, we will speak about it some minutes later when we discuss the positions about NFLD and its role in pathogenesis. One of the mechanisms of production of neurotransmitters, it is also due to the role of gut microbiota. Fermentation of non-digestible substrates like dietary fibers and endogenous intestinal mucus, which supports the growth of special microbes that produce short-chain fatty acid. It is one of the most important roles of normal bacterial cells, commensal bacterial cells. We know 3 types of these short-chain fatty acids, such as acetate, propionate, butyrate, and its ratio in normal situation is 3:1:1. And so the most important, the most abundant are acetates. Approximately 95% of all short-chain fatty acids are produced rapidly, and they are absorbed by colonocytes, and only 5% may be secreted in stools of our body. Acetate, what is the role of acetate? It is produced by the most part of gut anaerobes. It is produced by some types of firmicutes, Eubacterium rectale, Eubacterium hallei. It is for synthesis of propionate. And butyrate is produced as rule by Clostridium butyricum, Fusobacterium nucleatum, and Faecalibacterium prausnichi. Acetate, as the most abundant short-chain fatty acid, is essential metabolite of growth of other bacteria. It's not stay in bowl of gastrointestinal tract. It reaches the peripheral tissues, and where it is used in cholesterol metabolism and in lipogenesis, and also may play a great role in formation of central appetite regulation. Propionate is also, when it is produced in gastrointestinal tract, it is transferred to liver, where it is regulate process of gluconeogenesis. I wanted to speak and to tell in details about function of butyrate, because it is one of the most famous and the most important components of factors in progression of NFLD. Butyrate is the main energy source for human colonocytes. It may provide and induce apoptosis of colon cancer cells and can activate intestinal gluconeogenesis. This butyrate is one of the mediators which may control presence of oxygen in gastrointestinal tract. And we spoke that the most part of bacterial cells, normal bacterial cells, are anaerobes, and butyrate controls this process of some positions with anaerobical conditions in gastrointestinal tract. And as a result of this situation, it may control and prevent process of dysbiosis of microbiota. Butyrate promote tight junction function and intestinal integrity. It is one of the component which is disturbed in the situation with NFLD. Butyrate, by promoting of these glucagon-like peptides such as hormone, it acts on beta cells and provides secret of insulin. And the previous speaker told in details about action of this hormone. Butyrate is the most important anti-inflammatory mediator. It reduces number of pro-inflammatory cytokines. It prevents the progression of inflammatory responses to the systemic circulation. By strengthening of intestinal wall, intestinal barrier, some toxins and for example, lipopolysaccharide components will not penetrate into bloodstream and provide inflammation in the liver. We spoke about education of education of immune system, and in this case, butyrate is also activate regulatory T cells in the intestines and thus reduce pro-inflammatory signaling pathway. Butyrate may inhibit pro-inflammatory macrophages also. And it may stimulate rapid increase of production by an immune system of neutrophils, basophils, and eosinophils as the component of innate immunity. And we spoke about strengthening of gut barrier, so butyrate decreases the entrance of lipopolysaccharide layer into blood circulation and reduce inflammation. So we spoke in details about NFLD as a disease and hepatic manifestation of metabolic syndrome, which is characterized by hepatic fat accumulation, and previous speaker spoke in details about this situation. And I also want to say about the role of pathophysiology of NFLD as multifactorial, involving ecological, genetic, and metabolic factors such as limited physical activity, high energy intake, and disbalanced diet. Of course, situation with pathophysiology of NFLD is related to sometimes with antibiotic therapy and microbiota dysbiosis. And we should speak about situation with dysbiosis. So what is the dysbiosis and what may be according to NFLD? We spoke about gut-liver axis. And we showed this bidirectional relationship between gut and liver. And of course, change of normal composition of gut microbiota will provoke disruption of this gut microbiota and gut microbiota homeostasis. And what is dysbiosis in the cases of NFLD? Of course, it will be loss of beneficial microbiota. It will be increasing of pathogenic bacteria. It will be changes of metabolic activities and decreased of microbiota variety, reducing of diversity and disruption of intestinal barrier. And all these alterations may be as a result of dysbiosis and as a result of this gut-liver axis bidirectional relationship. So what types of bacteria are enriched in NFLD? You see here this list. So the most important, of course, the most abundant are bacteroidetes. These bacterial cells, which are gram-negative, they are enriched in NFLD. Of course, we may speak about Lactobacillus, Proteobacteria, Enterobacteriaceae family, about species of Escherichia coli, about Ruminococcus. And some types of bacteria, they are decreased in NFLD. So we spoke about change of ratio between firmicutes and bacteroidetes. And in this case, of course, we see that firmicutes are decreased in its amount, and proteobacteria and gram-negative enterobacteria, they are enriched in NFLD. And of course, this change of microbiota will provoke situation with dysbiosis, and the first one is, in this process we may speak about change of metabolism in the liver. And the first one, it is metabolic pathway, in which we may, as a result of dysbiosis, we may speak about some positions of production of change of metabolic reactions. So what is it? What is the first one? Of course, due to NFLD, as mentioned the previous speaker, we may see the change of structure, of composition of bile. So the first one, of course, it will be situation with dysregulation of bile acids. And as a result of it, of dysbiosis, we will see at first loss of microbicidal effect of bile. And one of the components of this antimicrobial activity is decreasing of number of immunoglobulin A as secretory immunoglobulins, which help to neutralize different antigens inside our gastrointestinal tract. And also this situation with losing of microbicidal effect will provide gastrointestinal bacteria translocation. And we may speak sometimes that presence of small intestinal bacterial overgrowth may be as a result of dysregulation of bile acids metabolism. The second position, which we may see in this slide, it is the situation with conversion of choline. And conversion of choline and dysregulation of choline metabolism will provide low level of synthesis of very low density lipoproteins. And as a result of it, it will be really problems with sometimes it will be risk for cardiovascular diseases. And also we don't forget that choline, it is the component of as phospholipid, it is the component of cell membranes and precursor for neurotransmitters such as acetylcholine. And in this case, dysregulation of choline metabolism will give us some problems with innervation inside the gastrointestinal tract also. And these positions is in the pathway of metabolic syndrome. Another component of metabolic syndrome is dysregulation in endocannabinoid system. So what's happened in this way, in this pathway? In situation with dysbiosis, we know that tryptophan, when it enters our body via food, in the situation with gut dysbiosis, it may be, there are three pathways for its metabolic reactions. Synthesis via synthesis of indole, via synthesis of serotonin, and via synthesis of quinine. When we have dysbiosis, we see that it changes tryptophan metabolism, and it will be situation with high level of production of serotonin, and energy expenditure will be decreased. And this pathway via quinine synthesis, of course, will provoke situation with high level of inflammation. And indole, due to indole, we may speak about its role in strengthening of gut barrier, of decreasing of inflammation and activation of lipogenesis. And if in this situation with gut dysbiosis we see in decreasing of level of indole, gut barrier will be disrupted, inflammation will be activated, and lipogenesis will be in low level. Next position for metabolic syndrome, of course, it is production of ethanol. In the situation with dysbiosis, we may speak about increasing of level of endogenously produced ethanol. In normal situation, when we have healthy body, alcohol is metabolized in liver by alcohol dehydrogenases. But if we have a large amount of endogenously produced ethanol in the situation with dysbiosis, when we see abundant of increasing of quantity of Escherichia, bacteroids, Bifidobacterium, Clostridium, and Klebsiella, we may speak about high level of production of ethanol, and it will provoke situation with some type of oxygenic stress. And this oxygenic stress due to high level of production of reactive oxygen species and nitrite oxide, which is also act as the toxic substances and may provoke distress, may provoke distress of hepatocytes by respiratory burst. So we spoke about the role of butyrate, and I told about quite high level of its importance in the situation with NFLD. And what's happened? In many researches, we may find information that as a rule, the level of butyrate is decreased, but another short chain fatty acids such as acetate may be increased in its synthesis. And if we have butyrate as a mediator for activation of macrophages and activation of immune cells as pro-inflammatory substance, of course, it will be situation with activation of macrophages and Kupffer cells in the liver. And as a result of it, it will be releasing of pro-inflammatory cytokines. We spoke that butyrate, it is pro-inflammatory, it is anti-inflammatory mediator, which improve intestinal barrier. And in the cases of dysbiosis, we may see increasing of permeability of gut barrier, especially to lipopolysaccharide components of gram-negative bacteria, and they will also provoke inflammation. So the role of butyrate is quite high, and we should speak about some dysregulation of this synthesis in the cases of dysbiosis. So we spoke about situation with metabolic syndrome. And the second pathway which we may find in pathogenesis of NFLD, it is situation with inflammation. So what are the reasons of inflammation inside the liver in the cases of dysbiosis? The first one, it is a situation with, at first, with change of properties of intestinal barrier, disruption of gut barrier will provoke its increasing of its permeability. But change, we spoke about change of ratio in favor of gram-negative bacterial cells, and we know that gram-negative bacterial cells in its cell wall, they have lipopolysaccharide components, which may act as endotoxin. And this endotoxin is one of the components of pro-inflammatory pro-inflammatory immune response. And as a result of increasing of number of lipopolysaccharides, we should speak about some situation with inflammatory pathway, and especially with activity of pattern recognition receptors. And the first pattern recognition receptors, according to its importance, of course, it is toll-like receptors. These toll-like receptors are located on Kupffer cells, on hepatocytes also, and they detect components of bacterial cells. At first, it is lipopolysaccharide layer. And if it is Kupffer cells, detection by these pattern recognition receptors and pathogen-associated molecular patterns, such as lipopolysaccharide layer, will provoke situation with activation of complement system and activation of C3A, C4A, C5A, and inflammation as itself. And also toll-like receptors will give us releasing of cytokines. We spoke about tumor necrosis factor alpha, interleukin-6, interleukin-1, and transforming growth factor beta. All these cytokines, it's not really as storm as maybe in some types of infectious. It's really releasing of cytokines, and we speak about low-grade chronic inflammations, which we may see in the situation of NFLD. And this component of inflammation, of course, it is one of the most important in progression and severity of NFLD. So how to if we summarize the situation with NFLD and gut microbiota, of course, we should speak about implication of many risk factors, that NFLD, it is implication of many risk factors. And how we may change this situation. Of course, the first one, it is lifestyle modification. And this lifestyle modification with the change of diet, with the presence of motivation according to loss of weight, according to increasing of physical activity. But we should not forget about mitigation of dysbiosis in gastrointestinal microbiota. And this situation of correction of dysbiosis may help us in progression of NFLD. So how to help our liver? We have some supplements, and these are prebiotics and probiotics. What is prebiotics? Prebiotics are a group of nutrients that are degraded by gut microbiota. We have two types of carbohydrates which may play a role of prebiotics. The first one is fructooligosaccharides, and the second one is galacto-oligosaccharides. We know that these carbohydrates, as rule, they present in food in small quantities, and that's why it's necessary to use as supplements in order to feed bacterial cells and normal bacterial cells, normal flora, because they may produce short-chain fatty acids, and we spoke in details about the role of these short-chain fatty acids. Probiotics, these are live bacterial cells and maybe yeasts or molds that we use with food, and it may present in fermented milk products such as yogurt or kefir, and sometimes it's really help us to control and to prevent situation with dysbiosis. And this is illustration of probiotics, and this is illustration with of situation of probiotics. So due to using of probiotics, we improve barrier gut barrier. We improve conditions inside gastrointestinal tract, and as a result of it, it will be a change of diversity of bacterial cells in favor of firmicutes, and gram-negative bacterial cells will be maybe will not survive in the situation with normal flora of probiotics. This is situation with probiotics. So colonization of microbiota will give us some success in not only prevention but as rule in situation with prophylaxis and the treatment of NFLD. So thank you. My report is over.
Hello. Hello, could you hear me? I hope it's hearable. Hello, let me start my talking. First, I'd like to thank my previous colleagues to a very lovely and useful lecture. And today I'm talking without the burden of non-alcoholic fatty liver disease in Kyrgyzstan and in the world. Content of my talking will include the prevalence of non-alcoholic fatty liver disease, comorbidities which associated with non-alcoholic fatty liver disease, and situation in Kyrgyzstan, of course, and the world, and diagnosis and some points about the treatment. Non-alcoholic fatty liver disease is the most common liver disease, the second most common cause of liver transplant, and the leader among indications for transplantation in person under 50 years of age. Alcohol is the first indication, and non-alcoholic fatty liver predisposing factor to primary liver cancer, even hepatocellular carcinoma may develop even by passing the stage of cirrhosis. You know, many diseases like chronic viral hepatitis and alcoholic fatty liver also may pass the stage of cirrhosis, but non-alcoholic fatty liver disease is not about non-alcoholic fatty liver disease, can directly go into the developing of hepatocellular carcinoma. And most people with fatty liver have normal enzyme value like ALT and AST level even maybe less than 40. Mostly non-alcoholic fatty liver more common in people over 45 years, and okay, sorry for interrupting. It's I think because of internet connection. I'm continuing. If you look at the prevalence, you see prevalence, worldwide prevalence of non-alcoholic fatty liver disease is about 25%. It varies from 13 to 30, and prevalence its advanced stage as non-alcoholic steatohepatitis in general population is between 1.5 to 6.5. We know that patients with type 2 diabetes mellitus more predisposed to non-alcoholic fatty liver disease. Among them, prevalence of non-alcoholic fatty liver reach 55%, sometimes vary from 30% to 80%. And prevalence of non-alcoholic fatty liver disease among children also growing, and among children it's about 8%. In 2015, our research group, STEPS, identified the risk factor of non-communicable disease in Kyrgyz Republic, and we investigated population at the age of 25 to 64. We found out risk factors of non-communicable diseases. Basically all diseases associated with metabolic syndrome have common risk factors, and we found that arterial hypertension, hypercholesterol level, smoking, small fruit and vegetable consumptions, obesity, low physical activity, excessive salt consumption spread very large. And non-alcoholic fatty liver disease is not only a comorbid pathology, but it also increases the risk of death and risk of malignancy. You see risk of cardiovascular death increase more than risk of cardiovascular death increase more than 40 times, and risk of malignancy, like liver malignancy, increased 4 times, stomach malignancy 3.5 times, pancreas also about 3 3 times increasing. Similar investigations came from Turkey, our Turkish colleagues. They also find out association of non-alcoholic fatty liver disease and its advanced steatotic hepatitis with risk factors like hyperlipidemia and obesity and metabolic syndrome, hypertension and diabetes. By 2030, experts predict growing of non-alcoholic fatty liver, and especially will grow the NASH, non-alcoholic steatotic hepatitis. And it's very important to solve this problem, to solve this problem today. You know, there is no non-alcoholic fatty liver disease in international classification disease 10 version, but 11th version will start from 1st of January in 2022. There are all forms of non-alcoholic fatty liver, even includes the unclassified form. And do we screen? Unfortunately, we have different recommendations from different organizations, but all of them, they're saying there is no systemic screening, but most of them highlighting the risk groups like obesity, type 2 diabetes, and dyslipidemia and metabolic syndrome. And in Kyrgyzstan, my team for the first time published guideline of non-alcoholic fatty liver disease. And we are also putting our recommendation, high risk groups like obesity, type 2 diabetes, and dyslipidemia, and even metabolic syndrome. Non-alcoholic fatty liver disease is disease of exclusion. For this, we should exclude the alcohol intaking, drug-induced fatty liver disease, any viral hepatitis, and other form of damaging of liver. Primary non-alcoholic fatty liver disease associated with metabolic risk factors and of course components of metabolic syndrome. That's very important components of metabolic syndrome. One study demonstrates the prevalence of severe fibrosis depends on the number of metabolic components. The greater number of metabolic components in patients, the higher the frequency of level fibrosis. Here is interesting phenotype which called lean non-alcoholic fatty liver disease. Not all obese people have non-alcoholic fatty liver disease. Not all people with non-alcoholic fatty liver disease are obese. Prevalence of lean non-alcoholic fatty liver disease varies from 3 to 30%. It might be higher in Asian countries. Asian, due to part of differences in body fat to muscle ratio, are particularly susceptible. Visceral fat, it's because of visceral fat, and visceral fat in the total pool of adipose tissue is 7 to 15%, but it plays a critical role in the development of insulin resistance. Visceral fat is saturated with cytokines and free fatty acids, and unfortunately, body weight, BMI, and waist circumferences do not reflect the amount of visceral fat. High prevalence of lean fatty liver disease, more likely in Asian countries. As you see in diagram, you see in Korea, in South and North Korea, and in Japan, and in China, there are high level of lean non-alcoholic fatty liver disease. 17 years observation of patient in United States, they revealed the non-obese non-alcoholic liver disease carries a higher mortality rate than obese non-alcoholic fatty livers. They compared obese and non-obese fatty liver disorders, and you see among the non-obese population, we have increased all causes of mortality because of cardiovascular diseases, and also because of cancer, and of course because of other reasons of death. Another 7-year prospective community-based cohort study from Sri Lanka also showing the development of comorbid metabolic diseases similar to the risk of non-alcoholic fatty liver with obesity. And also the PNPLA3 variant showed an association with lean non-alcoholic fatty liver disease in the study population. Another study of lean cohort, which includes more than 5,000 individuals, compare lean individuals without non-alcoholic fatty liver disease and with non-alcoholic fatty liver disease. They found significantly increased diabetes rate, increased high cholesterol, increased hypertension, and in the fully adjusted model, non-alcoholic fatty liver disease in lean was independently associated with an increased risk of all-cause cardiovascular mortality. Fatty liver disease usually asymptomatic, majority discovered by chance, often an incidental finding by finding of bright liver on imaging or abnormal liver test or finding of hepatomegaly. It's represented by asthenic syndrome, weakness, fatigue, any sleep disturbances, or sometimes it may be dyspeptic syndrome. But day by day, growing of scarring process in liver cause developing of portal hypertension syndrome, and will be may develop also hepatocellular failure syndrome. Non-alcoholic fatty liver disease is diagnosis of exclusion. Of course, at the beginning, we should exclude the alcohol intaking, but you should know that alcohol thresholds are oversimplified because the duration of significant alcohol exposure, drinking patterns, and individual susceptibility all play a role in alcohol-induced liver damage. For example, only similar dosages of alcohol for one person, it may be normal. For another person, it may have toxic effect. In our guidelines, we also leave this range of taking alcohol. For men, it's less than 30 grams per day. For women, less than 20 grams per day. If we talk about the non-alcoholic fatty liver disease, of course, on the second place, we put exclusion of drug-induced hepatitis. We should exclude taking of such drugs which cause the hepatocyte necrosis, which cause the cholestasis, which cause steatosis, fibrosis, or tumor. Of course, we also should exclude the autoimmune liver diseases. When we can suspect developing of autoimmune disorders, we suspect when gamma globulins level in serum proteins is high, which induce us to provide the immune globulin G examination, and then we check anti-nuclear antibodies, and of course we can check the antibodies to kidney microsomes, anti-smooth muscle antibodies, anti-mitochondrial antibodies, and etc. We should exclude the chemochromatosis. Chemochromatosis, of course, at the beginning, we should exclude by observation, a simple observation, by providing the physical examination of patients, and then detection of serum iron level, iron saturation of transferrin, also total iron binding capacity of serum, ferritin, and providing the genetic study, and if needful, of course, morphological examination of liver tissue. And next step, of course, we should exclude the Wilson's disease, detect the serum ceruloplasmin level and urinary copper expression, genetical study, and of course, examination by an ophthalmologist to detect the Kaiser-Fleischer ring. And one of the important point is exclusion of viral hepatitis B, C, D, and exclude alpha-1 antitrypsin deficiency. One, we have suspicion when increased alpha globulins level in serum proteins, and then it induces us to check up the alpha-1 antitrypsin level and provide the other like genetic study of mutation in gene A1 and AT. primary providers of fatty liver diagnosis, of course, is ultrasonography. Ultrasonography shows us phenomenon of attenuation of echo signal, difficulty in visualizing the architectonics of the liver and diaphragm. Unfortunately, changes visualized with steatosis more than 30%. So we could use another imaging methods like transient elastography, like acoustic pulse wave elastography or MR elastographies. And last one, in spite of very high cost of equipment, research takes a long time. It's very high advantages like high accuracy in obese patient. Of course, we have also provide some tests which based on combination of serum biomarkers, and these biomarkers search us how much fat and how much fibrous tissue, how much scar, how much fat, and sometimes how much activity of this process we can provide this. We know that fibrosis stage, the strongest predictor of liver-related adverse clinical outcomes in patients with non-alcoholic fatty liver disease. That's why so critically important to control stage of fibrosis. Controlling stage of fibrosis, we could use some scores. For example, here is most common non-alcoholic fatty liver score like NADAFL fibrosis score, FIB score. We just put the biomarkers and can get a level of fibrosis. For example, first yellowish color on the top, you see this score give you information about how much fat accumulation, intensity of fat accumulation, and in bottom shows you how much scar or fibrosis debris. Other investigations, because base of pathogenesis of non-alcoholic fatty liver disease, this is insulin resistance, you should check up on the fasting plasma insulin level and C-peptide. A value of more than 18 microunits per milliliter is regarded as basal hyperinsulinemia. And HOMA-R test, also very important, value is more than 2.27, indicates insulin resistance. And CARO index is a ratio of plasma glucose to fasting insulin level. If the index less than 0.33, it also gives us information about the insulin resistance. Liver biopsy remaining to be a gold standard, but unfortunately, only less than 5% of our patients agree with providing biopsy. And treatment goals very simple. We should treat impact on risk factors, also insulin resistance correction. Another goal is oxidative stress therapy, fatty hepatosis, treatment of fatty hepatosis, liver fibrosis therapy, and of course, therapy of associated clinical conditions. On the first place, located diet recommendations, create an energy deficit of 500 to 1,000 kilocalories to lose weight, 0.5 to 1 kilogram per week. Of course, we recommend our patients avoid consumption of simple carbohydrates, decrease taking of cholesterol content foods, and reduce consumption of red meat, and of course, reduce consumption of glucose and fructose. Coffee, unfortunately, here is coffee. Drinking at least 2 cups of coffee per day has a protective effect. All recommendations, all guidelines recommend us using Mediterranean diet. Of course, we should decrease consumption of glucose as well as fructose, especially fructose, because glucose and fructose absorbed by body in different ways. If glucose in our body absorbed by formation of liver glycogen and then passed to other tissues. And fructose has different mechanisms, mainly contributed to the de novo lipogenesis and sometimes may generate the uric acid. We also studied effects of fructose-rich diet in low and high altitude areas. Why low and high altitude areas? Because, you know, Kyrgyzstan population, 85% of Kyrgyzstan population lives at more than 1,500 meters above sea level. 42% lives above 3,000 meters above sea level. We provided this investigation on animals, and we keep them in fructose-rich diet during 10 weeks in high altitude and low altitude areas. And we have some reports, like in the high altitude group of animal on a diet enriched with fructose during 10 weeks, statistically low ALT level than in low altitude areas. And when we checked pre and anti-inflammatory cytokines, tumor necrotic factor and interleukin-4 in high altitude animals on a diet enriched with fructose has a higher value than in low altitude areas. And the level of pro and anti-inflammatory cytokines statistically significantly correlated with cytolytic syndrome, hypercholesterolemia, impaired synthetic function against the background of liver hypergosis. Those investigations, this is my own investigation, and I also I want to share with you, and also if you wish, you can join to such kind of investigations. I think in your country also providing like these researches. Observation study during 6 months shows 6 months of keeping of Mediterranean diet shows dramatically improving of steatosis. You see great frequency of grade more than 2 steatosis decreased in 80%, and even 20% we have resolution of non-alcoholic fatty liver disease. Diet is very important. For example, you see here study during 1 year study, and losing of 10% of weight give us very good results as steatosis improvement in 100%. Full steatosis improvement, NASH resolution, non-alcoholic steatohepatitis, inflammation resolution, 90%, and even fibrosis regression in 81%. Physical activity is also very important. Regular moderate aerobic exercises lasting 150 to 200 minutes per week. It may be walking, or it may be swimming or cycling, just to move. For those people who couldn't make themselves to keep diet, couldn't make themselves to doing any exercises, and have high obesity, of course, for those we are recommending bariatric surgery. And here you see improvement of in liver fat after bariatric surgery. If you look at in 6 months, significant fat pretty much gone away. You see from 31% to 3% decrease. Of course, we should recommend our patient avoid alcohol intaking. And this will be should be comprehensive therapy, comprehensive changing of lifestyle, comprehensive changing with the using of exercises. You see there are many organizations, guidelines. They're all till now continuing to research this non-alcoholic fatty liver disease, managing of non-alcoholic fatty liver disease. But pharmacological therapy, there are no drugs with proven efficacy and safety for non-alcoholic fatty liver disease today. Numerous studying now providing nowadays, and there are lots of different potential mechanisms. Depending where you are, you may have different treatment targets, like targets related to insulin resistance, targets related to lipotoxicity, to inflammation, to fibrogenesis, And for example, semaglutide, liraglutide, those kind of medicines, they mostly use for decreasing weight, and of course, also they continue to use in diabetes, in type 2 diabetes mellitus, or another obeticholic acid, this substance on the third phase of trial, and galectin has anti-scar, anti-fibrogenetic effect. Numerous studies providing nowadays all these substances in phase in third trial. What we have today and our protocols and our guidelines, we remain the metformin for patient who is suffering from diabetes to type 2 diabetes, and pioglitazone, but this medicine is sometimes limited. We are also providing in our country pharmacological correction of obesity by prescribing of orlistat and liraglutide, and we are doing pharmacological correction of hyperlipidemia. We are controlling hyperlipidemia by using of statins and sometimes, if needful, ursodeoxycholic acid. We are also providing antioxidant treatment. You should know that general patients with non-alcoholic fatty liver disease have lower blood level of zinc, copper, vitamin E, vitamin D, and vitamin E. But vitamin E using is limited because of we couldn't prescribe for patients who is suffering from second type of diabetes mellitus and who is suffering from steatohepatitis. We just can use for patient who has non-alcoholic fatty liver, and of course surgical treatment of obesity used in our country. What about ursodeoxycholic acid? Many guidelines, they remain this ursodeoxycholic acid only for treatment of primary biliary cirrhosis and for dissolution of gallstone. In our recommendations, we put this ursodeoxycholic acid because our patients are still losing weight, and you know that losing weight, this is good condition for formation of gallstones, and to prevent the formation of gallstones also we use ursodeoxycholic acid. And what about anti-hyperglycemic drugs for second type of diabetes? Some drugs, they are limited by developing of cytolysis syndrome. For example, if ALT level increase more than 2.5 times, we couldn't use pioglitazone or acarbose. If in liver develops a scarring process like cirrhosis, we couldn't use metformin using, and liraglutide using, of course, limited. But without limitation, we could use any DPPI-4 receptors inhibitors, and of course insulin. For DPPI inhibitors, we use DPP4 inhibitors, we use sitagliptin in our clinic. And here is clinical situations and which drugs we use if we have obese non-alcoholic fatty liver disease together with changing of lifestyle, and we are providing of exercises. Also, we may prescribe the orlistat, or we may prescribe the liraglutide. With second type of diabetes, it's dependable on liver function. We already discussed it. If associated with hypercholesterolemia, of course, we put statins and sometimes ezetimibe. And non-alcoholic fatty liver disease with intrahepatic cholestasis syndrome, we put also add ursodeoxycholic acid. And if we have association with the bacterial overgrowth syndrome, we use probiotics. Sometimes, of course, we may use sometimes antibiotics. And let me make some conclusions. Non-alcoholic fatty liver disease is part of multi-system disease associated with metabolic disorders. In addition to the classic non-alcoholic fatty liver disease phenotype, there are phenotypes such as lean non-alcoholic fatty liver disease and fatty liver in people living at different attitudes, which requires close attention of clinicians. Weight loss and lifestyle changes are the cornerstone of non-alcoholic fatty liver management. Development of treatment for non-alcoholic fatty liver disease continues, and the future going to be multiple medications associated with diet and exercise. Thank you for attention. Also, I want to say I want to thank organizers for good opportunity for us. Unfortunately, I couldn't see your faces, your facial expressions, and feeling myself as talking with a dark screen. Is anybody should close the session?